Auzubillahiminash-shaytanir-rajim. Bismillahirrahmanirrahim. This is Noman Ali. Today I am here to present my final year project presentation on the topic optimization of biogas produced from the campus waste. I was working on this project along with my teammate Sajjad Ali under the supervision of Dr. Farid Hussain Mangi and the co-supervision of Engineer Shakil Ahmed Jiskan. Special acknowledgements to Dr. Abdurrahim Channa for his continuous guidance and support in our final year project and review paper. This is the table of content that we are going to present today. The introduction, methodology, results and conclusion, future suggestion, and references. First of all, the introduction and the problem that we have identified. From these pictures, we can come to know that the problem that is being portrayed here is the food waste. The problem, food waste, the biomass are one of the biggest problems being faced by Earth right now, which are causing devastating problems day by day. The only thing that we know about the food waste is we are dumping them uh, in the landfills, and this is how we are treating them. But this is not a proper solution. We have to go for another solution that not only provides its benefits but is less hazardous for the earth, because the landfilling is causing the pollution and other problems for earth. Now, here are the biomass techniques, processing techniques. The biomass can be treated with two major process techniques, the thermochemical and the biochemical. There is another one called the combustion. Combustion is simply the burning of the food waste, the biomass. And we know that by burning them directly, we are creating the pollution by ourselves. Now, the thermochemical processing are such processes which takes place in the presence of fire or ignition, like the pyrolysis, hydrothermal, and the gasification. The pyrolysis and hydrothermal are the processing techniques which takes the biomass or the food waste as an input and provides the biochar or the bio-oil or gas as an yield or output. The gasification provides the gas or biochar as a yield. But we can see the thermochemical processes are working on the ignition or the burning of the woods or the fuels. So these are again causing the problem for the earth. These are not green solution. We have to move for the green solution. The biochemical processing has two main fields, the anaerobic digestion and the fermentation of the biomass. The anaerobic digestion provides the biogas or methane as an output or a yield, while the bioethanol is obtained from the fermentation process. So here is a comparison between the four main categories, the four main processing techniques. We can see the combustion works in the temperature range of 60 to 160 Celsius. The pyrolysis works on 400 to 800 Celsius. The gasification works on the temperature greater than 700 Celsius, while the anaerobic digestion works in between the temperature range of 40 to 55 Celsius. We can see the 40 to 55 Celsius is easily achievable throughout the world, while these temperature ranges require some median, some ignition, or other things consistently to maintain. The feedstock type, uh, the input that we provide to these techniques, process techniques, are the dry and the tough waste, or the coal for the combustion, the hardwood and the softwood for the pyrolysis, the wood, sludge, crop and the agriculture residues for the gasification, while we can use the food waste, the manure, agriculture waste, or the biogas, mostly the moist food waste, to the anaerobic digestion. The main thing about the anaerobic digestion is that it accepts the food waste which has uh, at least 70%, maximum 80-90% of the moisture content in them, which are not easily acceptable by any other processing technologies so far we have available, and the products that we have already discussed. Now, why anaerobic digestion? We have already gone through this, that the anaerobic digestion is simply the sequence of processes that works in the absence of oxygen, and they biodegrade the manure, food waste, or these things in the absence of oxygen. The main thing about the anaerobic digestion is that it can accept the highly moist food waste up to 90%, and the output that is obtained has the rich calorific value and the full of methane, and it promotes the green entrepreneurship. As a byproduct we get fertilizers and so on. Now, these are the process mechanism of the anaerobic digestion. First of all happens the hydrolysis, acetogenosis, and the methanogenosis. In a biodigester, anaerobic digester, we give the feedstock along with water in the ratio of 1 by 1 normally. So first of all happens the hydrolysis. What hydrolysis does, it breaks down the polysaccharides present in the food waste to the monosaccharides. Like the protein is broken down to the amino acids, the carbohydrates are broken down to the monosaccharides, the lipids are broken down to the long chain fatty acids. Now comes the acidogenosis. What acidogenosis does is breaks down those monosaccharides and creates the acids. We can see the volatile fatty acids, alcohols are formed by the acidogenosis process, and there comes the formation of hydrogen in slight ratio and the carbon. Then comes the acidogenosis. Acidogenosis combines the acids and the hydrogen, and the hydrogen molecules and the CO2 molecules are formed. In the end, the methanogenosis. In this process, the methogens present at the temperature range maintained are mixed with the acids and the carbon dioxide, and the methane and the carbon dioxide are formed naturally. Now, these are the achievements throughout our final year project. This is the uh, review paper that we are going to publish soon with the help of Dr. Abdurrahim Channa, inshallah. And these is the IEEE SCONEST 2022, which was held at Sakarya University. And our this topic, optimization of biogas production from the campus waste, won the first prize at the abstract presentation. Now comes the comparison of the different studies on anaerobic digestion. We took 6 most recent papers as a base and we worked on them. Now we can see the process type and the technologies. All of them works on the anaerobic digestion. The process types, some of them work on the single stage, few of them work on the 2 stage, and one was of 3 stage. The process type are decided on the basis of the pre-treatment of the food waste. We are not into them right now. Now we can see the fifth and the sixth author has used the food waste. So is the first one author, but it has used the sludge, and it has used the pre-treatment method. We can see the process parameters. Uh, this author has used HRT of 80 days. HRT stands for hydraulic retention time, means it has left the food waste for 80 days to produce the methane, which has the biogas with a concentration of 62% of the methane. This author has left the biodigester for 29 days, and the biogas that was produced had the methane concentration of 64% only, and so on. Now we have found a loop here. We aim to reduce the HRT and increase the methane production. So these are the aims and objectives that I have concluded throughout by the study of the literature. The food waste quantification and its characterization at the campus, since it's a campus study, and the optimization of the anaerobic digestion parameters, for instance, temperature, loading rate, retention time, and using the central composite design. So these are the 3 parameters that we are working on: temperature, hydraulic retention time or HRT, and the organic loading rate or OLR. The 3 temperature ranges that we have chosen: 40 degrees Celsius, 45 degrees Celsius, and 50 degrees Celsius. We have to optimize one of them. And after that, the loading rate 150 grams, 250 grams, and 200 grams. The retention time 20 days, 25 days, and 30 days. So this is the breakdown of our study, the scope of our study. First, we have to go through the characterization of the biomass by using the proximate analysis and the ultimate analysis. Then comes the optimization study by using the central composite design and the anaerobic digestion. The methodology. The first objective was covered here, the campus food waste survey. The food waste survey was divided into the two parts. First one was observing the participants, and the other one was visiting the kitchens of Sakarya University. Now we took 19 anonymous participants. We have not mentioned their names throughout. There comes the gender of them, observation place. We have observed three places: boys hostel 1, boys hostel 2, and the cafeteria of Sakarya University. We shared a meal with them and we observed whether they left their food in the plate at the end or not. And after our survey, we shared the result with them, and with their consent, we have placed these results here. And this is the study in which we have visited Boys Hostel 1, Boys Hostel 2, and the cafeteria of Sakarya University in order to find out what type of food waste is getting generated at the campus. So this is the thing that we are concerned about, the food waste from the rubbish bins, that we have the potato peels, chicken bones, cooked rice, cucumber, and spring onions, tomatoes, lemons, and so on, the fruits also. Now, this is the continuation of the first objective. We have been conducting the survey of all these three places, and as a total, we came to know that more than 1000 kilograms of the food waste is getting generated at the campus from these three places, where 700 people eat. And it is a rough approximation of 0.05 kg per person. Now, the characterization of the biomass. We go through the proximate analysis and the ultimate analysis. We have used white rice, potato peelings, and the cabbage as the feedstock for our experiments. That is why we have done the proximate and ultimate analysis of these three food waste. Now, the proximate analysis tells us the moisture content of the feedstock, while the ultimate analysis tells us about the composition of the elements like the carbon, hydrogen, oxygen of the feedstock. Now, to be sure, we know that anaerobic digestion accepts the food waste with the higher moisture content. So we need to be sure that the food waste that we are going to use in our experiment is high in moisture. Now comes the second objective, is the experimental design or the central composite design approach. We have already discussed that we are working on three different parameters: temperature, organic loading rate, and the hydraulic retention time. We have defined their ranges. We will be observing the experiments on three levels of temperature. First one, 40 degrees Celsius, 45 degrees Celsius, and the 50 degrees Celsius. The organic loading rate of 150 grams, 200 grams, and 250 grams. The hydraulic retention time of 20 days, 25 days, and 30 days. We have used Design Expert version 13.0 for the continuation of this study. This table was generated by the software. It has generated 17 different random experiments from these data provided. These are coded values. We will show you what does that mean. Now see, these are the factors. The factor 1 is temperature, the factor 2 is organic loading rate, the factor 3 is hydraulic retention time. In the experiment 1, the temperature minus 1 indicates 40 degrees. We can show you there. Now, what we have to do, we have to replace this 40 on the place of this negative 1. We came up with another table here. Now, these are 17 experiments that are generated through the central composite design. Like the first experiment says that do an anaerobic digestion of 250 grams organic loading rate for 20 days at 40 degrees Celsius. The experiment 3 says that carry out anaerobic digestion of 200 grams organic loading rate or the feedstock for 30 days on 45 degrees Celsius. Likewise, in the last experiment, 17th experiment tells us to carry out anaerobic digestion of 200 grams of feedstock for 25 days on 50 degrees Celsius. So these are the 17 experiments that we have carried out physically here. Now to carry out them, we have to make a temperature controlled bath here. So this is the experimental approach. We know we have to maintain three different temperature ranges: 40 degrees Celsius, 45 degrees Celsius, and the 50 degrees Celsius. Some of five of the experiments were defined for 40 degrees Celsius, the five other were defined for 45, and the rest of five were defined for 50 degrees Celsius. This is the experimental design that we have chosen. This is the container, and where we have to place our experimental bottles or the biodigester, because we were using 1.5 liter water bottles as digesters, and we inserted the feedstock in them, and they were maintained in the temperature controlled environment. So this is the ESP32 connected here, and this is the temperature sensor which was used to monitor the temperature throughout the days. And this was the temperature control rods or the geyser rods suspended here, which were electrically active for 24 hours for the continued 90 days time period. And this is how the experimental approach were designed. The experimental approach, we collected the feedstock in this manner. We found cooked rice from the cafeterias of Sakaraibi University, and the peelings of different fruits like mangoes, bananas, and the potato peelings from the cafeterias and the hostels. We wetted them and then we used them. This is how we made our setup. This is the geyser rod that we have used. It had thermostat, and it has the tripping system by default. This is how we maintained five different experiments at a time, because five experiments were to be carried out on 40 degrees Celsius and likewise. This is how you can see we were maintaining the temperature, and in the pyrometer you can see 46 degrees Celsius was maintained in this temperature controlled bath. Now the monitoring of the temperature control system. Now we monitored the temperature throughout the day for the whole 90 days period of our experiment. We were to maintain 40, 45, and 50. This is the graph of 1 day of maintaining 45 degrees Celsius. You can see there is very less uh, difference. The minimum trend line is 40 to 44.2, and the maximum trend line goes to 45. So the temperature was maintained between them. Now this is how we collected our biogas samples. In this digester, when the date approaches, we used to inject the blood bags, and the biogas that was produced in them was transferred here. Now coming to us the results. So after carrying out 17 experiments, we went through the gas chromatography of all the experiments of all the biogas samples that we had collected. Now you can see in the second experiment, we have achieved 75.8% of the methane in the biogas. Now the second experiment was conducted on 50 degrees Celsius to 50 grams, and for 20 days. Likewise, you can see in the experiment 15, we have achieved 76.18 percentage of the methane in the biogas, and there is a variation in the different samples because the temperature was maintained differently, the organic loading rate was different, and the hydraulic retention time was different for all the 17 experiments. Now, this is the comparison of the temperature and the HRT on the production of the methane. These plots were obtained through the help of Design Expert 13.0. Now we can see here, there is the temperature of 40 degrees Celsius, and here is the temperature of 50 degrees Celsius. Here the hydraulic retention time is 20 days, and here is the 30 days. We can see the 3D graph that at 40 degrees Celsius, 20 days, we had 55% of the methane in the biogas. But when the temperature range was enhanced to 50 degrees Celsius, and 50 degrees Celsius, again on 20 days, we received 75% of the methane. So the effect of temperature is significant. Now in this graph, we will observe the effect of the temperature and organic loading rate on methane. Again, at the 40 degrees Celsius, 150 organic loading rate, and 40 degrees Celsius, 250 grams organic loading rate. We can see there is no slight any difference available here, and 55 to 60% of the methane content was present in the biogas. But when the temperature was increased from the 40 to 50 degrees Celsius, we can see the effect is clearly visible here. The biogas contains 70 to 75% of the methane in it. But at the 50 degrees Celsius, if there is 250 grams of organic loading rate, we have the maximum methane concentration in the biogas. In this graph, we will observe the effect of organic loading rate in the hydraulic retention dam on the production of methane. We can see on the 150 grams of the organic loading rate for 20 days, we have the same pattern that can be seen for the 150 grams organic loading rate in 30 days. Similarly, for 250 grams and 20 days here, and the 250 grams and 30 days, the trend is similar. From this graph, we concluded that there is not significant effect of the organic loading rate in the hydraulic retention time on the concentration of the methane in the biogas, while in the previous two charts, the temperature effect was insignificant. Now, this is the statistical analysis. We can see the effect of temperature is significant. The p is less than 0.05, which signifies that the temperature is significant, while the organic loading rate in the HRD, their p value is greater than 0.05, which means they are not significant. This is the polynomial equation of the production of the biogas with methane concentration is obtained through Design Expert 13.0, and these are the optimum conditions that we have achieved throughout the project of 17 experiments. And we concluded that on the 50 degrees Celsius for 20 days and 250 grams of organic loading rate, we can achieve the maximum concentration of methane in the biogas from the parameters that we have designed so far. Now this is the confirmation of the model. We can see all the 17 experiments and their methane concentration, the predicted and the actual. The predicted methane yield was provided by the software. The software said after running the experiment number 1, you will be getting at least of 57.94 concentration of the methane in the biogas, and experimentally performance we achieved 57.101, which is quite similar. In the experiment 2, the software concluded that we will be getting 76.08 percentage before even running them physically here, and we achieved 75.8 percentage. Likewise, we can see this in these two experiments, we achieved the maximum methane concentration, and there is a slight difference, which means the error margin is negligible. Now these blue areas shows that the methane concentration was very poor in these experiments, while in these red experiments the methane concentration was rich. So in conclusion, we can say that desirability approach produces a combined optimum condition as follows: the temperature of 50 degrees Celsius and the OLR of 250 grams with 250 ml of water, because 1:1 ratio of the feedstock and water is taken, and the HRD of 20 days, which produces the biogas with a methane concentration of 76.2%, which is the maximum. The coefficient of variance is 2.5 of our whole experiment. which indicates that the model can be reproduced easily anywhere. The food waste survey concluded that more than 100,000 kgs of the food waste is getting generated at the campus from two boys' hostels and one cafeteria. And the temperature control system was designed here, in which we conducted all the 17 experiments. The future work or the suggestions. The successful completion of our study can be implemented at the campus for the clean energy generation. The retention time can be further optimized by the exposure of the pretreatment, usage of the industrial sludges as a catalyst, maintaining pH, and continuous stirring processes, and so on. The idea can be used along with the data present in the literature to do the comparative analysis of the different AI algorithms like ANN so that the process of the AD can be geographically optimized for the Pakistan. That's it. Thank you so much.